All right. I had a little cheat sheet I put together to introduce our guest here, but I think I'll just go out of memory because I can't seem to find it. But anyway, this is Dr. Charles O'Brien. He got his bachelor's degree in microbiology at the University of Oklahoma, and then obtained his Ph.D. in microbiology and immunology at the same university in 1990. Did some postdoc work at Yale and their Department of Cell Biology, if I recall correctly, and then came to Arkansas and joined on with the UAMS in '94, was it?
Exactly.
And now teaches internal medicine and
a variety of things.
A variety of things, yes. And I remember that you were also the director of the mouse transgenic facility there.
Correct.
And so anyway, join me in welcoming Dr. Charles O'Brien.
Thank you, Todd. If I wander around a little bit, can everybody hear me okay?
Yes.
Yeah. I'd like to wander around a little bit. Thanks, Todd, for the introduction, and convey my thanks to Anne. You see, I realize she's not here today for the invitation to come and speak to you today about this topic of genome editing and CRISPR-Cas. We'll get into the definition of exactly what those things mean here a little bit. I think the reason that Anne asked me was I think she initially contacted one of my colleagues in the transgenic facility, and just to try to get somebody to speak with you and to maybe lead a discussion about these issues that has direct experience with it. And so we do. When we make genetically modified animals at UAMS, we use CRISPR technology now to do some of that work. And so not just for animals, but also for cells in the lab, we're using this to do a variety of things. And we'll get into some of that in just a little bit. But I think that the reason that people want to talk about this is really highlighted by these two magazine covers right here. One of them pretty recent, the other one's not so much. But this idea of modifying human genes in order to accomplish various goals. One would be to maybe cure disease or limit disease susceptibility, perhaps. Or, I guess, a really popular notion that's being discussed a lot these days is designing humans, actually germline editing of human genes. We'll talk a lot about exactly what we mean by germline editing in a little bit. And that has a lot of ethical and societal implications. I'm certainly not an expert in any of that aspect of it. I'm a technical person. I use gene editing and other molecular biology tools to address scientific questions. But certainly, we can discuss anything that the group is interested in talking about. Although I'm certainly not an expert in those areas of ethics. So overall, today these are our objectives. Hopefully, at the end of our discussion today, you should be able to describe the process of gene editing and the components of the CRISPR-Cas system. And that way, when you hear about things on the radio or you read about them in the magazine, or you get into a discussion with friends or colleagues or family, you'll have some knowledge about how these things work, at least at a general level. You won't be able to necessarily go into the lab and get it done, but you'll have a general idea of how things work. And then hopefully you'll be able to recognize some of the potential benefits as well as the risk associated with use of this gene editing technology. And one of the, I think, hopefully a take-home message today is that despite all of the hype and news and in some cases perhaps it's going to be hysteria about the Steiner babies and human gene editing, this technology has tremendous potential and is already being used to accomplish a lot of good things. Certainly there are potential downsides to any new technology, but I hope that we can recognize some of the very beneficial things that gene editing can do for us. So here's a brief disclaimer. Although I am employed at the University of Arkansas Medical Sciences, I'm also a scientist at the Veterans Administration. All the views and opinions that I express today are my own and are not those of the institutions that employ me. And also, today's discussion is only for informational purposes. Nothing we talk about should be used to make healthcare or financial decisions based on anything that any information I have about how this technology works. So we probably have a fairly diverse crowd. This is the first time I've actually spoken to non-grad students or scientists about this topic. So I've tried to change the way I usually talk about it in order to appeal to a broader audience. And I'm sure that most of us are familiar with the basic tenets of biology and perhaps molecular biology. I thought I'd just touch on a few key points so that we're all kind of on the same page before we get started. And during this discussion and throughout the discussion, please, if I say something that's not clear or you'd like a little more clarification on, please just stop and we can elaborate on that a little bit more. Okay? So, the central dogma of molecular biology is really important to understand when talking about gene editing, that all organisms, every form of life on the planet has a DNA-based or RNA-based genome, nucleic acid-based genome. And most of all eukaryotic, all non-viral genomes, like everything that's all bacteria, all forms of life that aren't viruses, have a double-stranded DNA genome. All the genes that make up that organism are encoded in double-stranded DNA. And for that, and almost all organisms start out as a single cell. In case of sexually reproducing organisms, two cells come together, a sperm and an egg, and you get a single cell now that has those two nuclei that fuse together, or two genomes, one from the father and one from the mother that fuse together. Now you have a cell, a single cell that has two sets of chromosomes. And that single cell now starts to express the genes that are in those chromosomes to make the proteins that make up that organism. And the proteins are what do the job. The collagen in our skin is a protein. The enzymes that help digest our food are proteins. So proteins are kind of like the workhorses of the cells in our body. But all of the information to make the proteins, of course, is encoded in the DNA. And for that DNA to get turned into all the information in the DNA to get used to make protein, you have an intermediate molecule called RNA. And that's the RNA. There are many types of RNA. This type of RNA is called the messenger RNA. It has the message to make a protein. The message from the gene is transcribed into RNA, single-stranded RNA, and that gets translated into protein. The protein does the job. So you go from this single cell that's just one type of cell, and it starts to divide and then differentiate all the different kinds of cells. We start as one kind of cell, and then we get liver cells, we get bone cells, we get blood cells, we get neurons for our brain, all these different types of cells. And all the information how to make these different cell types, of course, is encoded in DNA. But there are other types of RNA in the cell besides the messenger RNA. And that's going to be a little bit important for our discussion today. There are structural RNAs. RNAs that are actually used to help the messenger RNA get translated into protein. And there are small RNAs that we don't even know exactly what they do. There are lots of different kinds of RNA in the cell besides the RNA that's used to make protein. And we're going to need to know about one of those RNAs. So any questions about this kind of general genes to RNA and protein? So we're going to start out by talking about CRISPR itself. Just kind of get that out of the way, and then we're going to come back. We're going to leave it. We're going to come back to it in a minute. But where did it come from? What is CRISPR-Cas? So the CRISPR-Cas system was originally identified in a bacteria, the bacteria that causes strep throat, called Streptococcus pyogenes. And what it is essentially, it's a naturally occurring thing that occurs in all bacteria, or almost all bacteria, and it's essentially an immune system for bacteria. So you're probably familiar with our immune system. We've got an immune system that has two basic parts. It has innate immunity, things like our skin barrier and enzymes that we secrete to kind of defend ourselves against invaders. But we also have an adaptive immune system. When you get immunized for the flu, they give you some of the proteins that are made by the flu virus, not an active virus, but just the proteins made by that flu. And your body recognizes those and starts to make antibodies against those proteins, so that if you see the virus, now you can block that virus from entering your cells. So you adapt to that antigen, that immunization, the vaccination, by making antibodies to that. That's an adaptive immune system. And we do that all the time. We adapt to things that we encounter and develop antibodies against them so that we're protected against them. Well, bacteria can do kind of the same thing. So like human cells, and animal cells, and plant cells, bacterial cells can be infected by viruses as well. And many of the viruses that infect bacteria are double-stranded DNA viruses. So, as I said, all forms of life, except viruses, have a double-stranded DNA genome. Viruses come in many flavors. Some of them have a double-stranded DNA genome. Some of them have a single-stranded DNA genome. Some of them, like HIV, have a single-stranded RNA genome. None of that really matters, except that the viruses that this immune system for bacteria works with, it helps them recognize the genomes of double-stranded DNA viruses. What happens is when a bacteria is infected, so this is the surface of the bacteria here, and this is a virus. They call them phage. If you have a virus that infects a bacteria, it's called a phage. When the phage injects double-stranded DNA genome into the virus, or the virus injects into the bacteria, one of two things can happen. It's going to start to replicate, and it's going to kill the bacteria, and it's all over for that bacteria. But if the bacterium survives, what it can do is it can cut up a piece of that viral DNA here and incorporate it into the genome of the bacteria, into these little blocks here. And each of these different, this represents a strand of the DNA of the bacteria. And each of these colored blocks is a fragment of a different piece of viral DNA that this bacterium or its ancestors were exposed to in the past. And it keeps that in it as part of its genome. But it doesn't encode a gene. It encodes a small piece of this viral genomic DNA. Well, how can that possibly help this bacteria? What can happen is the bacteria has evolved over the eons a set of proteins that can now transcribe this DNA into RNA, into something called a guide RNA, and then other proteins that complex with that guide RNA that then can recognize double-stranded DNA from that virus if it sees it again, and cut it and inactivate it. And if you cut that incoming viral DNA, it inactivates it and it can no longer be infected. So it protects the bacterium from this virus in the future. And that's what the CRISPR system is. It's ongoing. The different kinds of bacteria have different versions of this, slightly different versions of the system. The one from the Strep pyogenes is the most widely understood and the one that we use in the laboratory the most, that we've kind of adapted. So we'll talk in a little bit more about how we've now taken the components, or some of the components, of this bacterial adaptive immune system and changed them so that we can use them as tools in the molecular biology laboratory to do things. Any questions about yes?
How do you specifically, what tool do you use to cut that in and place it in that strong one?
So we don't do that at all. This is a naturally, in the bacteria, this is a naturally occurring process. It has enzymes that will cut up the incoming viral DNA and then insert that into the bacterial genome DNA. That's not something we do. That's something that bacteria do naturally all the time. But how we take advantage of that, I'll come to it in just a moment.
So you're not inserting enzymes either. It's using its own enzymes.
It's using its own repair machinery and DNA replication machinery to incorporate that viral DNA, that foreign DNA into its genome. So this is all happening, or has been happening for millions and millions of years in all different kinds of bacteria all the time. It's going on around us all the time, still today. This is just a naturally occurring process that we've learned about over the last long time now. So it's been since the recognition of these small pieces of viral DNA in bacterial DNA was first reported in 1987. So we've been sequencing DNA for, since about 1975-ish, so a long time. And so some of the first things we could get our hands on to sequence were with bacterial genomic DNA. So about 10 years into it, in the mid-1980s, we started seeing these repetitive elements in bacterial genomic DNA that didn't seem to encode proteins. Nobody knew what they did. But they coined the term CRISPR about 2002 to describe these sequences that people found in the different bacterial genomes. Nobody knew what they did. And then as time went on, people, microbiologists in the lab, slaving away, figured out, okay, They finally figured out it was an adaptive immune system for bacteria. And then they got this all figured, the adaptive immune part, they figured that out. The idea came about this some time ago, back in 2005. They finally nailed it down in the early teens. And it was only in about 2013 when people figured out, well, maybe we could use this tool to do molecular biology in other cell types, non-bacterial. And then, it was first demonstrated in 2013. So it's only been about 5 years that this technology has been around for use by us in the lab, as opposed to something bacteria. So, yes.
So does the CR CRISPR stand for clustered repeat?
That stands for, I've got one here. It stands for, I don't even know, clustered regularly interspersed short palindromic repeats. So those are those fragments of foreign DNA that are found in bacterial genomic DNA. That's what they stand for. They're clustered together, and in between those viral sequence, there's a repeat element, and that's what the repeat stands for. And they're interspersed between these viral DNA sequences. And they happen to be a thousand.
So you're not altering the virus that you infected with at all?
We're not going to do anything.
You're just using that, picking out viruses at what you
Well, what we're going to do, what some really smart scientists, both out in California and out in Boston, figured out was that they could take components of this system from the bacteria, specifically one protein and one RNA. The protein's called Cas9. It is shown here in this outline in green, just a diagram of it. And the small guide RNA. That's the only two components of the system you need. And what those do is that the protein, the Cas9, binds to the RNA. They form a complex, a very stable complex. And what that complex can do is it can bind to double-stranded DNA, and it can just start reading, scanning along. It's like a train going down the tracks. And then what it's doing is it's looking for a sequence, a specific sequence. It's looking for the sequence GG. So remember there are 4 bases for DNA, A, G, T, C. And it's looking for 2 Gs in a row. And every time it finds 2 Gs in a row on either strand of DNA, remember our DNA is double-stranded. Anytime it finds a GG, it stops. The complex stops and it says, do I have a match to the 20 nucleotides that make up the 5' end of this RNA? So just the first 20 nucleotides of this RNA, does the DNA that I'm scanning, does it match in sequence that sequence? So RNA and DNA have almost this, they can have the same sequence. Just remember, RNAs have, DNA has GATC, RNA has GAUC. U's and T's have a match. A can bind to T or A can bind to U. So it just uses Watson basic pairing to say, do I have a match to my RNA sequence, the guide RNA sequence? And if it does, it will cut. The Cas9 is a multifunctional protein. It can bind to DNA, it can scan DNA, and it can cut DNA. And that's the key thing, is it cuts DNA. And it makes a cut, a double strand, it cuts both strands of DNA about 3 nucleotides upstream from this NGG sequence. And that's all it is. If that piece of double-stranded DNA happens to be an incoming piece of viral genome, bam, it's cut and inactivated. End of story. End of infection. That's what it normally does. But what these scientists realize is sometimes it's a good, if you can make a double-stranded DNA cut in the genomic DNA of a cell, like a eukaryotic cell, a cell from a person or a plant or an animal, you can do things with that. That can be the beginning of a tool or a process that you can use to do things. And we're going to talk a lot more about that in a minute, but it's important that you understand these components. It's a relatively simple system. Yes?
How exact does that 20 nucleotide match have to be in order to cut?
That's a really good question. Pretty close. Now, if you happen to have some mismatches toward this end of it, you can tolerate 1 or 2, sometimes 3 mismatches near that end. The closer you get to the cut site, the more accurate it has to be. Now, that brings up this question of when people are using CRISPR-Cas9 for gene editing, so-called off-target activities, cutting where it's not supposed to cut. And that does happen. It happened a lot earlier on, but It depends on the sequence. Sometimes some sequences are very specific, some are not. And people have been working on the versions of the Cas9 protein that are much more specific. And that's one of the goals these days, is to come up with a Cas9 guide RNA complex. It will only cut with an exact match. And I think we're just about there in terms of specificity. We're very close. They've got new mutant versions of Cas9 that won't cut unless there's an exact match.
This is, I guess, a classic problem in computer science, string matching. Say, here's a long character string, here's a short one, look for the an occurrence of this short string in this longer one there. There are a bunch of algorithms for that, including some very efficient ones now that are much better than what people do on just sort of intuitively. Do they understand what the algorithm is that does that?
This is
pattern
really
matching. Is it like anything in what string pattern matching is?
Probably not. It's a relatively straightforward model. The analogy that I use is actually pretty accurate, where this complex binds and scans the entire genome. It's reading everything, and only stopping when it recognizes that GG, and then it unwinds some of the DNA and says, "Do I have a match to my sequence here?" So it's basically testing the entire thing, 20 nucleotides at a time. So it's not very efficient, but in any given nucleus, when we're using this as a tool, there are hundreds of thousands of these complexes scanning your genome, and it does it pretty quick. So it actually turns out to be relatively efficient.
Parallel lines.
Yes, it is a highly parallel system, absolutely.
So who made the Cas9 protein?
Who made it?
Yeah.
The Cas9 protein is a naturally occurring protein from the Streptococcus pyogenes bacteria.
Oh, okay. Yeah, no.
What we've done is we found a way to make that. We basically cloned the gene for Strep pyogenes, so we can now express Cas9. We can put it anywhere we want. We can even make a protein in a test tube and put the protein in cells. The key thing was the guide RNA. So in Strep pyogenes, this guide RNA encodes homology to a lot of viral genomes. So that doesn't do us any good as a tool for molecular biology. In the lab, it's really easy to change this first 20 nucleotides of the guide RNA, just using standard molecular biology techniques. And now we can make it cut anywhere we want. We've kind of hijacked the system, and we change what sequences it recognizes to cut. So we can make it cut anywhere we want. Well, with one constraint, that sequence has to be upstream from the sequence in GG. So we can't cut just about. We can cut just about anywhere, but not exactly anywhere. Fortunately, there are Cas systems from other bacteria that have different recognition sequences. Different, instead of a GG that I recognize an AA, or a GGG, or something like that. And so you can typically find one that will recognize just about any sequence. So before we talk more about using gene editing, let's think about some of the classic reasons people want to do gene editing. That's human genetic disease. So I think everybody's probably familiar with sickle cell disease, which is a very difficult condition in which the red blood cells of homozygous carriers of this condition, under certain conditions, the hemoglobin it misforms in red blood cells of these people and causes them to form a very abnormal shape, a sickle shape, and they're really larger, a lot larger than normal red blood cells. So they have a tough time going through the small capillaries. They clog up, those areas become ischemic, tissue starts to die. It's incredibly painful and debilitating disease. This disease is caused by a single base change in one gene. And if they're homozygous for it, that's when they have a really bad hormone. But it's in the alpha hemoglobin gene of people. And if we can fix that, if we can change that one base pair back to the normal base pair, you can cure these individuals of the disease. You would make a normal hemoglobin. So if you were thinking about types of gene therapy, There's a lot of approaches you could use for gene therapy. You could go in and try to fix the gene that's mutated, like in sickle cell disease. Or you might try to introduce a good version of the gene someplace else, like randomly integrate it into the genome or give it to them exogenously. Well, that wouldn't work in this case. If you gave an individual with sickle cell disease, if you gave them a good copy of the gene someplace else in their genome, that's not going to help them. They're still going to have this bad version, still making the abnormal hemoglobin, and it's still going to cause the problem. You've got to find a way to fix that particular bad gene. They're meant in this normal place in the genome. And until recently, that's been a huge challenge. We have not had a way to do that. And that's one thing that gene editing, if it works well, can give you the opportunity to do. Another common human disease is severe combined not common, but one that people are aware of, is a severe combined immunodeficiency. These individuals can't make any adaptive immune cells. They can't make B lymphocytes and T lymphocytes. The B lymphocytes are the lymphocytes that make the antibodies. So these individuals can't, if they get immunized with a flu viral vaccine, they can't respond to that, and they can't make antibodies to the vaccine, or they're not protected. They're not protected to any of the organisms that they encounter. You remember, you probably don't remember, when I was growing up in the 70s, they had this movie about the bubble boy. Boy had to live in a bubble because he had no adaptive immune system. That's what these individuals have. They can't make any antibodies. And so that's due to the deficiency in an enzyme called adenosine deaminase. Now, in this case, you wouldn't have to go in and fix that bad copy of the gene. You could give them a good copy of that gene someplace else in their genomes by randomly inserting it, and you would cure these people. So that's another type of gene therapy that's a little less specific. You just have to supply them with a little bit of what they're missing. Another disease like that would be called the one where you can't clot, hemophilia. They're just missing a little bit of one of the clotting factors. If you gave them a foreign gene that encoded that missing factor, and they made just a little bit of it, you would probably cure that person. You don't have to necessarily go in to their exact spot in the genome where they've got a mutation and fix it. And so before gene editing came along, what could we do? How could we do different types of potentially gene therapy, or do anything with DNA? So this is where my job comes in. I change DNA all the time, not in people or plants, but in rodents. And one of the ways we do that is something called transgenesis. It's a funny sounding word, but all it means is taking foreign DNA and inserting it into the genome of an organism. That's all it is. And it's a relatively crude but simple but effective process to use in mice. In many cases, to study different diseases or processes in animals, or to create animal models of human disease, you want to insert a synthetic gene into the mouse. And the way we do that is we create, in the lab, in test tubes, we create a synthetic gene that does something we want. Make a protein in perhaps it does normal thing. And we take that DNA and we inject it into the fertilized egg from a mouse. And then that fertilized egg, I don't know if you can see it, but there are two small, so there's a big holding pipette holding it on the right, and that's got just a little bit of suction in it to hold on to that fertilized egg. And then on the left, there's a needle, it's a glass needle that's filled with DNA solution. And what my colleague will do is he'll inject that needle into the egg, and there's two small nuclei in there. I don't know if you can see them. They're really, really tiny. One came from the sperm, and one came from the egg. So they're called pronuclei because they haven't fused yet. There's one, and there's the other. Now, in a few hours after he does the injection, those nuclei are going to fuse, and then that cell's going to start dividing into all the cells that are going to make the embryo. So when we inject DNA into one of those nuclei, it incorporates into the genome and becomes part of the genomic constitution of that animal. The thing is, it's random. We have no control over where that goes in. We just hope that it goes into a place where it will be expressed and do its job. Sometimes it goes in, it's silent, doesn't do anything. But hopefully, every now and then, we can identify one of the offspring where that DNA is integrated, it makes the protein we want it to make, and now we've got a model we can use to study something in the lab. So we've been doing this since the early 1980s, and it works very well. But you can't use that to fix anything. Or you could do something like this in people, perhaps, but you could just introduce foreign DNA. You You could cure something like adenosine deaminase deficiency, but you're not going to do anything for something like sickle cell. Now, do we have a way to make very specific mutations in the place that we want to make them in a mouse? The answer is yes, but it's really complicated. We have to use something called embryonic stem cells. So has anybody heard of embryonic stem cells? So they have two really important properties. One is, if you take embryonic stem cells and you introduce them into a developing embryo. So down here on the right, you see something that's a little bit like what we showed earlier. Here's the holding pipette, here's the developing embryo, and here's a glass pipette. This one's a lot bigger than that needle that I showed earlier. Instead of injecting DNA, it's introducing embryonic stem cells. And what will happen is we get this embryo at just the right time, we put these foreign embryonic stem cells in there. They will incorporate into the developing animal and actually contribute to the tissues of that animal. And if it gets into the gonads of the animal, that will be in the germline. It'll be able to pass those cells onto the offspring, or the genetic information from the cells onto the offspring. So if you make a mutation in the embryonic stem cells, you can make mice that have that mutation or that change, that genetic change in their germline. And so the other property they have, besides being able to contribute to tissues of the developing embryo, is that they undergo something called homologous recombination. And that simply means that if we put in a piece of DNA that has homology to an endogenous gene, so this diagram here is of a gene that's in the genome of those embryonic stem cells, and this is a foreign piece of DNA that we make in a test tube that has homology to this region. And we use that homology to make that DNA go to the place in the genome, the DNA sequence that we want it to go to. And if we put something new in between those homology arms, it will introduce that change into the gene in exactly the spot that we want to put it. So you can make a very precise change. You could turn a mouse version of a gene into a human version of a gene. And you can use that to then study therapies for humans, but only use mice instead of, so we do that frequently. Sometimes we make drugs that will only work on the human version of a protein, but we don't want to, of course, test that in people. But we want to see if it works, so we humanize that gene in the mouse, and then we see if that protein or that antibody, that therapy, will work in the mouse before we go on to people. So this works for making very precise, very accurate changes in DNA, but for this to work, you have to have embryonic stem cells. And for most organisms, we don't have embryonic stem cells. We don't have cells we can grow in a culture dish in a lab that will continue to be undifferentiated and maintain that pluripotent state indefinitely. These mouse embryonic stem cells will do that. So they're really unique in this. We have that for a few species, but for most species, we don't have it. We don't have it, as far as I know, for humans. We don't have it for even rats. We don't have it for monkeys. I think the only species that I'm aware of that we have it for are mice and ferrets.
So what are they taking out of people's spines when they say taking stem cells out, then putting them back in after the chemotherapy?
Right, so there are different kinds of stem cells. A stem cell in general is just a cell that's some kind of progenitor that replicates itself. So it stays in an undifferentiated state and then can, under certain conditions, then become other types of cells. An embryonic stem cell is capable of becoming any cell type in the body. The cells you're talking about are called tissue-specific stem cells, or mesenchymal stem cells for the most part. And those have a much more limited potential. They can only become certain types of cells. They are pluripotent, meaning they can become many types of cells, but they're not omnipotent. That's not the right word. There's another word. Pluripotent means many, but there's a one that is, they can become any cell type in the body. Totipotent, that's it, thank you. Totipotent. So embryonic stem cells are totipotent. Most types of therapeutic stem cells are only pluripotent. Sometimes they only have a couple of options, a couple of cell types. They're more committed to a certain lineage. Yes?
So, you probably don't want me to bring this up, but the thing in 60 Minutes that everybody was watching like crazy, where they were talking about how they cured a small group of people of sickle cell.
Yes.
They were using pluripotent stem cells and introducing the new DNA into those?
Yes. So we'll come to that. Yeah, there's something called a hematopoietic stem cell, and it's a really very important and useful cell type in the human body and in all bone animals. And it is a stem cell that we can have good access to because you can get from the bone marrow. You can isolate them, and they reproduce like crazy. And so it's like this. If I were to, if they take my bone marrow, they can get a small sample of my bone marrow and isolate just a few hundred hematopoietic stem cells, keep them alive and undifferentiated in a culture dish. Now they can give me a drug or radiation treatment that will wipe out all of my bone marrow, kill it all. And so I can't make blood cells anymore. And if something doesn't, if I don't get autologous or heterologous bone marrow transplant in a few weeks, I'm going to die from bone marrow failure. So they can take those and give them back to me and save my life. Just a few hundred of them, theoretically, just a few dozen of them, can completely repopulate my bone marrow. It's just phenomenal. You can actually do that in mice. We have done that in mice a lot. So they have tremendous expansion potential, and they maintain their pluripotency quite well. So what they did in 60 Minutes, and what we'll talk about in a minute, is they can make genetic changes in those cells, very specific, and check and make sure that it's the correct one. They can then treat the person, like I said, ablate their bone marrow and give them back. So now the only cells they're using are the correct, the cells that are making the correct version of the gene they.
I use that video in my classroom because I have to keep it something simple enough that my kids can grasp.
I think it's the best example right now, is the sickle cell example. It's one of the first clinical trials that was used. There's a lot of ways they can fix that mutation. I think we'll touch on a couple of those in a little bit. But yeah, that's exactly it.
As long as we're on that, though, I think you said he used 500,000 in the bag that they injected back into.
Oh,
you have 500,000 of your cells in this bag. And also, this will she have to take anti-rejection drugs her entire life?
no.
She is
Those are her cells. The only thing that's different about those cells is that the gene that was mutated is now corrected. But all the rest of the proteins on that cell are, she recognizes them as her own. That's the great thing about it. It's like an autologous bone marrow.
So would you say this is the first time we, as humans, have actually been able to play God? And
I wouldn't say that.
No, that's pretty impressive to take somebody who's got a disease that will kill her early, no doubt, and very painfully, and turn that around and make her completely regular.
Yeah, but that's a tremendous accomplishment. It's vaccinations have been tremendous. Have saved, not just saved, of course, millions of lives or billions of lives, but improved the lives of just innumerable people. So I mean, there's lots of great accomplishments in science. I'm a big advocate of science, or I wouldn't do what I do. This is a big step for us. So we'll come. I think if we have time to talk about that, talk about how, or the things to think about in terms of, now that we can do this, should we do it, and how should we do it, and when should we do it? Those are really big questions that we're wrestling with right now as a society, globally. So back to gene therapy. So until recently, until the advent of the CRISPR-Cas era, the only way we could approach gene therapy, because we don't have human embryonic stem cells that we know will work, and the ethics of doing that kind of thing, was to do it kind of like a transgenic, kind of like a transgenic human. We could see for this one. This would be like with the adenosine deaminase deficiency. This approach would work, and they tried this. So they actually got it to work successfully. You can take those hematopoietic stem cells that lack adenosine deaminase from the patient, hematopoietic stem cells, and we can make a virus that has been inactivated. It doesn't make the viable proteins. It's an inactivated virus, but it can carry foreign genetic information. So we put a good copy of the adenosine deaminase gene in that viral genome, and we use that to introduce that DNA into the hematopoietic stem cells. The altered virus is mixed with cells from the patient. The virus gets in there, inserts a good copy of the adenosine deaminase gene just randomly someplace in the genome of these hematopoietic stem cells. We put them back in the patient, and now they start to make the enzyme. They begin to make functional lymphocytes, and the person gets now an adaptive immune response and is cured. Right? We did that without gene editing. That's just a crude way of doing it. Now, that sounds great, and it is great, it works, but there's one huge problem. Those viruses, when they integrate into the genome, they do it randomly, and sometimes they integrate near a gene that, when activated, will cause cancer. And that happened. In the first dozen kids they tried this in, 4 of them got leukemia because the virus integrated and activated the wrong gene. They said, okay, we can't do this anymore. And then we recently, a group in France thinks they have found a way to prevent that from happening. But still, people are very afraid to use this kind of approach to solve those kinds of diseases because you have no control over where the virus integrates. And so that really put the kibosh on gene therapy for about 15 years. Okay, can't do it. So, this is a slide from my favorite television program, which some of you may be familiar with. So in this particular episode of Star Trek, Spock and Captain Kirk have been thrown back into Depression-era New York City. And the only way that they can get back to the future is if Spock can create a computer that can access the information on his tricorder and get it back. But of course, it's really hard. There are no computers in 1932. And so he's got these vacuum tubes and copper wire. He's working hard in the basement. Captain Kirk is out running around with the Eden Keeler. And he comes back and says, Spock, how come you haven't got the computer working? He goes, look, Kirk, Captain, I'm working with the equivalent of stone knives and bearskins to create the mnemonic memory circuit. And a lot of people like me that watched this when they were kids grew up to be computer scientists and engineers. And in that field of computer science and engineers, the term stone knives and bearskins is used as a shorthand to describe a technology that is no longer that most is not the best for that particular purpose. If you want to use something that's up to date and appropriate, that you don't use this technology. And although transgenesis and homologous recombination are still good, we still use them, compared to gene editing, it's like stone knives and bearskins. It's a totally new set of technologies. They're transforming the way we do genetic engineering in animals, plants, and people. So what is gene editing? Now this is probably, if you don't get any other message or understand anything else we're talking about today, this is, if you want to understand gene editing, you need to understand this diagram. So the way gene editing works is, you start by creating a double-stranded DNA break at the place, at a very specific spot in the genome of the organism. And so that's indicated by that lightning bolt cutting both strands. Now, in any type of eukaryotic cell, whether it's a plant, a fungus, or a person, any kind of organism, that break will be repaired in one of 2 ways. On the left is a process called non-homologous enzyme, NHEG. And that's the most prevalent way that that break is going to get repaired. And machinery in the nucleus of the cell will recognize those free DNA ends and just glue them back together. And that sounds pretty good. The problem is, when it does that, frequently it will nibble back a few bases or add a few extra bases, creating a mutation. That's what we call, if it takes some away, it's called a deletion mutation. If it inserts some, it's called an insertion mutation. For short, we call them indels. Now, if that break happens to be in a region of DNA that encodes a protein, and you have indels that occur in a protein, its coding sequence, that's going to either disrupt the normal sequence of that protein, or more than likely, it's going to cause the protein to be truncated and non-functional most of the time. So indels in a protein coding sequence are usually going to inactivate that gene, and you're not going to get it's basically we call it a knockout, because you no longer make that protein. Now, the other thing that can happen is that if there is a source of DNA that has homology to the region near the break site, that other piece of DNA can be used to repair the break in a more precise fashion. And we call that other piece of DNA the donor template, and the process is called homology-directed repair, HDR. It's much less efficient, but when it works, it makes a very precise repair or modification of that sequence. And so what we can do is we can take this donor template and we can insert whatever we want here. And so we can decide where we're going to make the cut, and we can decide how we're going to change it when it's repaired. If we can select for cells that have undergone this process, we can make any genetic change in any sequence that we want. So that's a key thing to really think about. We can make any change in any genetic sequence we want, in any organism. Now that is astonishing. It's also very powerful. We've never before in the history of science had the ability to do it, but now we do. So what can you do with it? Well, this is just this is how to make that double-stranded DNA break, we use Crispr, just the way as I had described it before. We build a guide RNA that recognizes the sequence where we want to make the double-stranded DNA cut. Then we supply the homology donor that makes the change that we want to make, and we select for cells that have undergone homologous recombination. Or in some cases, we're just trying to inactivate a gene. Sometimes, if we want to make a mouse that is missing a certain gene, so we just make indels, and that works very well. It works so much faster and easier than doing the embryonic stem cell technology. That takes about a year. The embryonic stem cell one, we want to make a knockout mouse, a mouse that's missing a specific gene. It takes about a year and takes about $30,000. If you want to do it using CRISPR-Cas9, it takes about 8 weeks. It takes about $3,000. So it's just changed the way we do things dramatically. But see, gene editing is not new. Gene editing has been around for about 15 years. It's just before CRISPR-Cas9, we had very complicated, inefficient ways of making double-stranded DNA breaks where we wanted to make it. We had to use these other two versions of enzyme called zinc finger nucleases and talon nucleases. And for those to work, you don't have to just change the guide RNA sequence like you do with CRISPR-Cas9. With CRISPR-Cas9, for every new sequence you want to cut, you just change the first 20 nucleotides of the guide RNA, which is super easy to do. It takes about $100 and about 3 days of work in the lab to make that change. And a high school student. To make a new zinc finger or a new talon takes many, many weeks. I can't do it. I have to hire experts at companies to do it, and it costs tens of thousands of dollars. So people have stopped, well, for the most part, stopped using zinc finger nucleases and talon nucleases because CRISPR is just so much cheaper and easier. But the idea of gene editing has been around for a long time. And some of the first therapies for sickle cell anemia were gene editing not using CRISPR, but using just zinc finger nucleases. So, how do you use these things? Well, a variety of ways. In the lab, where we grow cells in culture, or we can get embryos from mice, we can inject the components of the CRISPR-Cas system into the eggs, just like we would to make a transgene. Instead of that needle, instead of having a transgene, we have a homology donor and the Cas9 protein and guide RNA. Just inject that instead, and it works. Or we can introduce those components into cells in a culture dish using some different scientific approaches to get DNA or proteins into cells. So that it's relatively easy. If you can get fertilized eggs from a species, you can do this. Or there's lots of ways that you can, using these transcriptional preparation or transduction, you can use that to get it into things like plant cells. So just about any cell type that you can get out and grow in a culture dish, you can do CRISPR-Cas9 gene editing. You just have to have access to the cells. And that's what makes diseases in which there's a hematopoietic problem so amenable to this kind of thing, because you can get the stem cells out, keep them healthy in the dish, change them, select the ones that have the change you want, and put those back in specifically. That really makes those kinds of diseases much more amenable. I think one of you, I heard someone mentioning something about cystic fibrosis. So cystic fibrosis is a single gene mutation in a membrane channel. The problem is, and it would be great to have a gene therapy for that. The issue there is we can't get the stem cells. So it's not like blood where you can just go to the bone marrow, take out the stem cells, and grow up in culture. You have, there are many different problems with the cystic fibrosis condition. The main one is the lungs, of course. The lungs, they don't secrete, the secretions in the lung are too thick, and it makes it very difficult to expel bacteria, and things build up, and it's very difficult. That creates fibrosis in the lungs, and then the lungs can't expand, the alveoli can't expand very well, and then they lose lung function. If you wanted to go in and repair that version of the cystic fibrosis gene, you've got to get to the stem cells in the lung, the epithelial stem cells in the lining of the lung, and make a change there. And right now, we don't have a way to do that. You know,
So we are
inhaling,
spending money on finding it.
yeah, that's
I work for the Cystic Fibrosis Foundation. There's some significant research that includes CRISPR-Cas, and I have a lot of other areas as well. So it's interesting to hear about it more.
the key will be if they can invent something, come up with a plan, grab some kind of inactivated virus that they can inhale, and then would take that CRISPR-Cas components into the lung of the person. The problem is that right now, we don't, the efficiency of the CRISPR-Cas gene changes aren't high. With hemophilia and embryonic and hematopoietic stem cells, we can screen all the different cells and find the ones that have the right change. We try to do it in vivo by putting the components into a live organism. We can't, if bad things happen in some cells, we can't select for that. So it's a real challenge. It's something other than hematopoietic stem cell right now. It's going to be a real challenge to figure out how to do that. Eventually, they're going to solve that, but right now, it's a big challenge. So, but any disease that happens in blood cells is a great candidate for gene therapy using CRISPR-Cas9 gene editing. But this technology is going forward outside the realm of human health in a huge way. So we can do protein clear injections to make cows, goats, pigs, all kinds of agricultural animals. And you can use it to make changes in those animals, to do all kinds of things. You can change the properties of the animal. You can use that animal to make human proteins for therapy, like human insulin, for example, made in goats and poultry. That's been done. Whatever kind of change you think would be beneficial in the genome of one of these important animals, you can make that change. That's going forward at light speed. An interesting question that came up in the process of doing that is, are animals that have been modified by gene editing the same as genetically modified? Will that be viewed as genetically modified by things like the FDA or the similar organization in the EU? And the EU came to the conclusion that they would be considered genetically modified. And that's really important because the majority of the public, both in this country and in the EU, don't like genetically modified. They think if it's genetically modified, that's necessarily a bad thing, and they don't want anything to do with it. And so that's going to have a big impact on the use of this. I had a conversation with some colleagues up at Fayetteville about, they do a lot of work with poultry companies in that part of the state, about they have this problem sometimes that they make these chickens that get so big, so heavy, that their femurs break and they can't use them. So I've got a genetic change that can fix that. I study bones, osteoporosis. So there's a very simple genetic change that we can make in chickens that would give them basically super strong bones that would never break. But they're not interested. You know, it would solve the problem, but they couldn't sell the chickens. Nobody would buy it. Nobody would eat it. So they're not interested in any genetic modifications like that. But if we could get the same modification by selective breeding or a randomly naturally occurring mutation, and that would be fine. But if I make that same change on purpose, no go. So, but it's essentially the same thing.
So, what are the chances that you can get a mutation that will cause the disease? Or is it just a matter of finding the right mutation?
Well, the chances are, if you find the right mutation, you can fix it. But it's essentially the same thing. So, it's, but it's essentially the same thing.
What are the chances that you can get a mutation that will cause the disease?
But it's essentially the same thing. So this is an article about the same group of people that you mentioned that was on 60 Minutes, where they have now taken some, they've done some clinical trials. They've corrected sickle cell anemia or treated it in one of two ways. In one group of patients, one study, they went in and tried to make they correct the endogenous exact gene to make that single base change go back to the way it should be, and they put those cells back in. And that's pretty difficult. The other way they did it, there's a way, you have many forms of hemoglobin. You have something called fetal hemoglobin and adult hemoglobin. The sickle cell gene is mutations in the adult hemoglobin gene. The fetal hemoglobin gene gets turned off early, shortly after birth. What they found the way to do is to inactivate a gene, making an indel, that allows the fetal hemoglobin gene to get turned back on. So now they've got a lot of good fetal hemoglobin that essentially takes the place of the sickle cell hemoglobin, and that accomplishes the same thing, just by kind of an indirect way. So both of those clinical trials have gone pretty well, and they're not approved as a therapy yet, but they are progressing toward that pretty soon. And remember what I said, if you remember what I said, that this technology has only been shown to work in eukaryotic cells for about 5 years. So we've gone just from initially getting the tool that would do the job to actually doing clinical trials in 5 years. That's phenomenal. I don't think there's anything like that. It's incredible. So if we can do it, of course, in agriculturally important animals, can we do it in primates? Very early on in 2014, just a couple of years after the technology was developed for this kind of use. These monkeys were made, and they actually did the opposite. They inactivated the genes that are important for an adaptive immune system in these monkeys. These two monkeys don't have an adaptive immune system. But it showed very early it worked in primates. They did this by pronuclear injection. And so if it works in monkeys and primates, of course it's going to work in people. And we know now, of course, that it does work in people. We'll talk about that in a minute. But as soon as those, before that, this most recent thing with the two female babies were produced, another group in China gene edited some human zygotes that were not capable of developing into healthy humans. They were pronuclear zygotes, which is a rare thing that happens. But they did it just to test would it work in human embryos, and it did. And they published this paper. This is something weird that happened. Typically, when we submit a paper for publication, it takes months and months and months to get it in press. And it's go through a huge review process. There's a lot of back and forth between the authors and the editors and the reviewers. This paper was submitted on March 30th, 2015, and accepted by the journal a couple of days later. That never happens. That's how weird things are getting with gene editing. And so when that paper came out, the world responded. The New York Times, a lot of news outlets made a big deal out of it. And scientists got together and said, we need to think about this. And so they convened, as they usually do, a conference and said, and they came out with a statement that we need to slow things down and think about it a little bit more, recognize the benefits, but also the risks. But this particular conference and statement, which came out in 2015, had no teeth, had no real statement. And as we'll talk about in just a minute, the response this time around has been a little bit stronger. So as you feel, probably everyone's heard about late last year, this scientist in China announced at the Crispr conference that he had genetically modified 2 human female babies so that they would not be able to be infected by HIV. Their father was HIV, and that was ostensibly the reason for doing it, so that he wouldn't be able to infect his children growing up in this environment. But it has created a huge firestorm of criticism for the scientists, for the Chinese scientific establishment, the Chinese government, and for science in general. I think that a lot of people are outraged that this is happening so soon when we cannot guarantee the safety of this approach. And these 2 children now, no one really knows. It has been confirmed, at least by the Chinese government. They claim to have confirmed the existence of these 2 children, and that they're their genes have been modified by CRISPR-Cas, but we don't know the long-term consequences. We don't know if, in addition to that change, other changes were made and what the long-term consequences would be. And they certainly had no say in the matter. So, a lot of ethical considerations when it comes to thinking about doing this at all. And if you're going to do it, how do you do it, and what constitutes a good reason to do it? So I'm not paying any attention to time. Where are we with the time?
3:30.
Not trying to wrap it up? Has anybody heard of a gene drive? A gene drive is something that, so a gene drive is an idea of a way of spreading genetic information in a wild population of organisms in a non-Mendelian way. So if I take let's say I have a goal of creating a population of mosquitoes. I want to wipe out the ability of mosquitoes to carry the organism causing malaria. So we know in a mosquito, we can make a genetic change so that it'll no longer carry the parasite. Mosquitoes will be fine. They can still do their thing, but they just won't be able to transmit malaria. That'd be a pretty good thing, potentially. But there's no, if I make that change to the mosquito and I turn it loose, it's that mutation is not going to spread very well. It's not going to spread very effectively using a traditional Mendelian genetics. It just doesn't have the power to do that. What a gene drive does is it overrides mammalian genetics. Now that version of the gene is selfish. It can copy itself from the haploid state to the diploid state, and now any organism that has that change can only create offspring that have that change, and they create offspring that only have that change, and so on and so on and so on, so that a genetic mutation can spread in a wild-type population. Sounds like science fiction. A guy named Austin Burke came up with this idea about 15 years ago, and there was no way to do it back then. He just said, this would be how we would do it. I mean, if we could do it, this is the kind of problem we could solve. But two scientists at MIT said, we figured out a way to do that using CRISPR-Cas system, and they made it work in fruit flies. So that the mutation that should have only been passed as a heterozygous mutation was passed as a homozygous mutation, exactly like a gene drive should work. So it's not just an idea, it actually works. So that you now have the potential of creating a gene drive, introducing it to wild, and having that mutation spread uncontrollably into the entire wild-type population, which we've also never had the ability to do before. And when the Zika problem came up a couple of years ago, some people said, well, maybe that our response to this should be creating a gene drive that will wipe out the ability of Zika carrying mosquitoes to reproduce, essentially eliminate the species on purpose to solve the problem. Which might be a good idea, might not be a good idea. I don't know. They didn't take that step. They've never come up with a vaccine that's pretty effective for Zika virus. So, as far as I know, this has been put on hold for now, but there's going to be other viruses spread by mosquitoes that are going to have to be dealt with. So, to end with, in summary, there's a lot of things that this technology can be used for. We've touched on some of these here. This kind of paints the big picture a bit. In my realm of work, and the two on the right, the animal models and epigenetic variation, we use this technology a lot in the lab to do very useful, important, what we think are relatively safe things. We create animal models to study human diseases, how they're caused, how to treat them, test treatments. We can test the function of genes in cell models in the lab. We talked about gene therapy, here called gene surgery, and drug development. The area of agriculture and material science, and we didn't talk about very much, but the material science is really an interesting area where they're now creating using CRISPR to modify a yeast to essentially make all kinds of compounds that could never be made before by anything. You can tune a yeast to make all kinds of synthetic materials that in the past were impossible to make because you couldn't create all the enzyme pathways necessary. Now we can. So that's a burgeoning area of material science in yeast and fuel production, in addition to foods with certain desirable traits. So I hope that provided you kind of an overview of the technology behind CRISPR-Cas9 and gene editing, with just touching lightly on some of the ethical and societal concerns that might be involved. Thank you for coming.
One of the suggestions I've seen, I think it was in New Scientist actually, for applications used in livestock. Because there are lots of animals like egg-laying chickens or milk cows where males are basically useless or practically so. Using the CRISPR-Cas9 to do away with males. Have you heard anything about this being done?
I'm not. Potentially parthenogenesis, a way to do parthenogenesis. I don't know.
Well, this was one of the suggestions as a potential application. And I didn't know whether anything along that line had been done. Because it's true, I mean, you know, roosters in an egg-laying farm are much used.
Well,
You might have to have one or two around, but mostly they're useful.
well, that's the way it's done. For some species, artificial insemination works really, really well, so you don't need that. But the old-fashioned way still works pretty well. I think most agriculture still have done that. But I don't know. I'm not familiar.
But you're saying that the only way it can be done is with a Y chromosome. You must use the Y chromosome to reproduce.
Or at least parts of it, probably. But this is also a matter that,
You
I
still
mean,
have-
birds don't have the same sex determination as mammals do, so it would be different for birds than it would for animals. They were going to do something.
I think animals still do have a Y chromosome. Is that not true? I don't. I'm not certain of that, but I think.
Yeah.
But I think the sex determination.
I think all chordates have the same. They're reproduced by sexual reproduction. Yeah, you're right. The Y chromosome carries very specific sex-determining genes, and you could probably tone that down to just the essential ones, but you'd still need it.
Right.
There's a line of insects that we raised at the Museum of Discovery. They're a type of walking stick, and they've been reproducing for quite a while with parthenogenesis. It's all females in the line. However, the problem is that the genetics degrade over time. And we've gotten to the point in this line that they're not reproducing well at all anymore. So it works within limits.
Yeah, when it comes to population health, diversities are really, really good.
Well, and I mean, in terms of selecting for male versus female offspring, it works. I mean, in mammals, you have the XY chromosome model, but like in reptiles, a lot of reptiles, you have a male and a female, but the determination as to whether or not the egg develops into male or female is based on the temperature at which they're incubated. And that determines which genes get turned on and off to make the offspring male or female. And, you know, something like that, you don't need to, you know, genetic is in a role. Just adjust the temperature.
I guess you can get male.
Well, no, you need to have a male to mate with the female, but the eggs that are produced, whether or not they end up male or female, it isn't an XY determination.
I just had a woman at my dance club, a young woman, say this: we no longer need the male to reproduce. And I just went, okay, I just let it go. I just can't debate things like that.
Well, there's a really, really good science fiction book that has something like that in it. Has anybody ever read the book 7 Eves? 7 Eves? Like Adam Eve,
yeah.
7 Eves? Oh, that's really
Give them another century or so.
You said that they did traditional type of gene therapy like in 1 year and $30,000.
Well, to make a genetically modified mouse, when we used ESL technology to make a knockout mouse, it took about a year, about $30,000. But we can do the same thing now. Really, we did this. We did it in 8 weeks from idea to actually having the pups be born, screened. It only took 8 weeks. And that just is.
How much is that?
That cost ran about $3,000. So about a 10-fold drop in cost.
So does this have anything to do with growing the human ear on the back of the mouse? Is that another field?
Yeah, that's.
You haven't seen that done
I
yet?
remember something about that, but I don't. That wasn't a transgene or anything like that.
Yeah, that's, no. know. I think they transferred that here to someone.
Wow. Well, one thing they're trying to do is they are trying to develop ways to grow human organs in things like agriculturally important species like pigs and goats and cows, which would be really a good thing, I think. And the way they do that is they have to get really good at making pluripotent stem cells from like be able to take some cells out of meat and get them to become pluripotent, and then find a way to program them to make a kidney, for example, and implant those into a pig. The problem with using porcine-derived tissues in people is that, of course, most strains of pigs have endogenous retroviruses. And they are, in a pig, they're kept relatively at bay. So you take those tissues out and you put them in a human, those retroviral sequences get activated and can cause oncogenesis, kind of like we did with the gene therapy in humans, and cause leukemia. So there's no way to get rid of all the endogenous retroviruses. Well, you could probably try to clean up the mouse genome using gene editing. And I think maybe that's what they're trying to do. They're trying to make it so that they can grow these human tissues in a pig, and then, but the pig has been modified so that it doesn't have this endogenous retrovirus problem. I've read a lot about that, but I know they are definitely trying to do that. The first step is they created mice in which they grew rat kidneys, and about a usable percentage of those are actually functional kidneys. So that's going to actually be a huge improvement for kidney organ transplant.
We're using pig valves for heart transplants.
Yeah, extracellular. Yeah, there's no live cells in there. Yeah, it's just extracellular matrix there. Essentially, just tendons.
Most of the way through here, you refer to Cas9. In your titles, you had just Cas, and I know there was a long search from the first time they discovered any Cas systems to come up with Cas9 as the best of the ones they've searched for. Do they use any of those?
Oh, yeah. There's a lot of other versions now that are being used. They have different properties. They recognize different sequences. Instead of the GG, they have different sequences as their seed sequence or where they start searching. None of them are as widely applied as the Cas9, but they're coming up. In fact, we're developing a procedure that I think will have to have both. We'll have to have one that uses Cas9 and one that will have to be different in order to, because we want to use different guide RNAs to do different things. I think we're going to have to bring in one of these other systems. So I think they're gaining popularity. Well, I appreciate you spending a little bit of your St. Patrick's Day hearing about something that I think a lot of people find a little bit disturbing, but hopefully has some real benefits.
I think that's already been proven.
In my view.
Except for the cost of doing that. I mean, you had to do every patient. Sickle cell, I bet it costs a fortune to do that right now.
They're thinking several millions of dollars.
Several million. The one they did successfully was several $1 million.
And that's a huge issue. So for cystic fibrosis, there are certain mutations that can be treated very effectively with a small molecule chaperone. It's basically a medicine you give to these individuals orally, and it essentially cures them. I think Vertex was the company that created it.
It's the OTR modulators that cause the protein development.
And what's the annual cost for that drug?
There are 3 now, and we're on the verge of one that will actually treat about 90% of patients. So it varies. It varies, but it's 300,000 and slightly more. It's very expensive. Vertex is doing quite well in the stock market. You're right about.
So, and I have friends that have developed monoclonal antibody-based therapies for rare diseases, and they all start around $300,000 to $500,000 a year to treat these people, which is unconscionable in my view. And it's so expensive. Arkansas was, I think, the last state to approve insurance or Medicaid funding for those patients, and there was an effort to deny them access to, or at least pay for. But no individual or private do that for very long for these people. So we got to come up with a different model if we want to. Otherwise, only the very, very wealthy people can afford to not only buy access to for their kids to certain institutions, they'll be the only ones that have access to these therapies.
We appreciate you coming out.